So this is from a book called Make It Stick, which is a really interesting book on how the adult human learns. And they presented a lot of case studies and neuroscience to back it up. But really, as you're going through your academic career, you are literally changing your brain. You are creating new neural pathways and strengthening those neural pathways. The things you don't think about on a regular basis weaken and die off. And so the more you think about something, the more neural pathways get laid down and strengthen up. And so that's why they say it is really not a good idea to just wait until, a few days before the exam and study. That won't do it. You need time to lay down those neural pathways. It does not happen in 24 or 48 hours. And so that's why periodic practice is a method to do that. All right. And so when I say studying a little bit every day, you are taking steps to lay down neural pathways in a consistent pattern that strengthen over time. And I don't mean study for hours on this class nonstop. It's best if you do short bursts of very active studying. And that can mean different things for different people. I really don't advocate just reading your notes from start to finish and then reading your notes from start to finish in mindless kind of pattern. You really have to get active with your study process. For me, that meant I couldn't study in the library because I'd fall asleep too easily. And so I would study in my room and I would walk around, I would talk out loud, I would draw on my whiteboard. I would use things in my room that were meaningful and personal to me and attach it to certain things I was learning. And so that when I would take the exam, I could just kind of picture myself studying and manipulating and holding those things that had meaning to me. It's kind of like building a memory palace, if you've ever heard of that study strategy. That's what some of the competitors that do those annual international competitions on memorization, essentially, where they can memorize a deck of cards that comes flashing at them and they can just do it. They use memory palaces in order to build that type of learning process. So I guess I was doing that before I even knew what it was. So I like to be very active when I learn, like physically speaking things out. I even act things out if it makes sense to me. And so whatever you got to do. And that may mean the library might not be the best place, but it just depends how you learn. So periodic practice arrests forgetting. Definitely. Testing yourself as you're practicing, not, you got to know what you know, and then you have to figure out what you don't know. And really the only way to do that is to test yourself, quiz yourself, go over and learn the material, and then shut your laptop and see if you can retrieve it. Think about questions that might be asked on an exam and think if you can answer it without looking at your notes. And so again, you got to do that before exam day so you can figure out, yeah, I'm confident on this stuff, I know this stuff, or oh boy, I don't know this stuff, I better brush up on it. If you don't do that before exam day, you're really gambling when you walk in on exam day. Okay, so use testing as a tool. Elaboration in your own words, being able to link what you're learning this semester to past content goes a long way. So starting to build that knowledge base, and the further along in your academic career you get, the easier that gets to be. And so as you get into your junior and senior years, it gets easier and easier to learn new information because you've built such a knowledge base that it's much easier to make connections and put things in your own words. And then this phrase, illusions of mastery, is something that I fell prey to when I was an undergrad student. I had to really learn how to study, learn how to learn. Rereading your textbook is really not a good idea. Rereading your notes over and over and over, just kind of mindlessly doing it, not a good idea. And so you start to think you know it just because it looks familiar to you. But familiarity does not mean you know it. And that really is illusions of mastery. I did this to myself too when I was an undergrad. And it took, a test, an exam or two for me to realize, ooh, I studied, but I didn't study in a way that created knowledge. It was just, I was familiar with things. And that's a different thing. If you're familiar, you certainly are going to have a hard time with any short answer questions. As multiple choice, you may be able to muddle through it, but not always. So being familiar with something doesn't mean that you know it. Okay, so think about that idea as well. Okay, I'm going to stop there, and I'll talk to you a little bit more over the next several lecture sessions to get you thinking about this. And, I encourage you to play around with it, even if you think you already know how to study. If there's a more efficient way to do it where you can spend less time studying overall because you're more efficient, I think everyone would like that, right? We all have really busy lives and you have a lot of studying to do. The other thing I really encourage you to do is have fun with it. I would create little sayings and silly little acronyms that would make me laugh. Maybe somebody else wouldn't think it funny, but if you can inject humor into your study, and they actually show laughter and humor helps you learn. The neuroscience is showing it. I don't know all the details on that exactly, but they're showing it to be true. So you spend a lot of time studying, you might as well make it fun and as painless as you can. So some things to think about. All right, we're going to get into the plant body. That's first, we got to think about that. So in this class, we're going to start with a little bit of plant anatomy, and then kind of get into diversity. So this slide set is concerning the plant anatomy, and we're going to zoom into cells first, plant cells, and then branch, kind of zoom out a little bit to get into the tissues of the plant body. So when you look at plants compared to animal cells, there's some major key differences, okay? So certainly the chloroplast is something that we don't have. I sure wish we did have that, because then we could just make our own food, but unfortunately we can't do that. So plants have chloroplasts. That's where photosynthesis takes place, how they make their sugars and get a lot of their ATP. But they also have mitochondria like we do to make their ATP, so that they are kind of special that way. Several ways to do that. You can see that some of the other organelles there as well, the nucleus and lysosome. So what I want you to mostly focus on is the idea of the chloroplast. We're going to think about that in more detail as semester goes. The other thing on there that we don't really talk about with animal cells is the vacuole, right? The vacuole. So the vacuole is really important to the plant because it is a storage, it plays a storage function. Okay, so it can store pigments, for example, to give certain colors to certain parts of the plant body. So pigments could be in there, enzymes, proteins. They even use the vacuole sometimes if they need to stuff some kind of toxin that they can't excrete. They'll stuff it into the vacuole and keep their other precious organelles safe. The vacuole also has another role that's really important because it can fill with water. That's the main ingredient. If you were to crack open the vacuole, it would be full of water. And if you have a nice healthy cell, that vacuole is going to be jam-packed with water to the point that it's pressing against the membrane of the vacuole and causing pressure. And so the Turgor pressure idea is what we're thinking about here. The Turgor pressure is the idea of water exerting. That's not very. I got an old one here. Turgor pressure. Can you read that well enough for right now? All right, I'll get a better one. Turgor pressure is water pressure, and it's the vacuole is hopefully doing its job of creating that Turgor pressure by all the water that it takes in. And what you end up seeing is a situation where in a mature plant cell, which is drawn here, the vacuole is the largest organelle, not the nucleus. I think if you take intro bio, you might have been taught the nucleus is the largest organelle. Well, they tend to focus on animals, forget about plants. So with plants, we have the vacuole as the biggest organelle. And so that jam-packed with water exerting pressure, it exerts pressure against all the organelles, all the material, which exerts pressure against the cell membrane, which exerts pressure against the cell wall. And so it's a way for the plant to have some rigidity and structural support just through water pressure. They don't have a skeletal system like we do. So they have to figure out a different way. They evolved a different strategy to fight gravity and have some structural support. Okay, so very, very important. The membrane, by the way, of the vacuole is given the name tonoplast. Tonoplast. That's the membrane. All right, so let me go back. So for this class, I'd like you to read up for sure in the book on the chloroplast, the nucleus, the vacuole, and the mitochondria. So make sure that you're thinking about those organelles when you think about the plant cell. And we'll come back to these as we work through some of the topics in class. By the way, the picture you're seeing on the left is an example of a plant tissue. This is actually the tip of a plant root. So we'll be getting into that as well. The cells and tissues. So let's go back to the cell picture. And so when you, when you look at the plant cell, there's a couple of other things I want you to look at. So let's look at the boundary of the cell. You see that there's a cell wall. And in this rendition, there's two cell walls. There's the so-called secondary cell wall, and then secondary cell wall is being shown there incorrectly. Just caught that. They got it reversed. Let me show you how it should do it. Roll. Okay, so I'll edit these slides because that's I'll like draw on it so that you can see what I'm. Whoops. All right, so this is a slide for the primary cell wall is being shown as darker tan band, and then the secondary cell wall is the lighter tan to the inside of the primary cell wall. All right, so when a plant, when the seeds are germinating and growing, all the plant cells in the plant body, as the plant is maturing and growing, all plant cells have a primary cell wall. Okay, primary cell wall. So we usually denote that with number 1 and 3 symbol as a superscript. Primary. That means it's primary. So primary cell wall, all plant cells have that. Okay. You see that compared to the secondary cell wall, it's a little thinner, but it is pretty strong in the sense that it provides nice structural support. Okay. Then the secondary cell wall you see there is being laid down to the inside of the primary cell wall. So we denote the secondary with the 2 superscript, the degree symbol. So the secondary cell wall, if a plant decides to lay that down, it will be a bigger cell wall, thicker, and it has slightly different chemical composition, which we'll talk about. Now, not all plant cells have a secondary cell wall. Only certain areas of the plant body will have that occurring. If that plant cell is being designed to go ahead and make a secondary cell wall, once the secondary cell wall is laid down and everything's good to go, the cell actually dies. The internal contents, everything goes away, and you're left with a hollow cell, essentially. It's dead. Now that may sound bad, but it's actually good, because certain parts of the plant body having hollow cells in those certain parts actually is a benefit and a really good strategy. So we'll talk more about that too, okay? So the secondary cell wall, if it's going to be formed, it's formed to the inside of the primary cell wall, and it is thicker and has slightly different chemical composition. So I'm going to edit this one so it's not showing that incorrect pattern of primary to secondary. Was it this one? Yeah, that one. Okay. Questions on that? Okay, well, let's think about the cell wall in a little bit more detail. Ultimately, yes. Yeah, yeah. And then the membrane will dilate. Once the secondary cell wall gets made, and everything's good, like the secondary cell wall is all intact and good to go, then everything inside essentially goes away, dies on. So the cell walls, whether it's primary or secondary cell wall, they both have cellulose. And cellulose, really, when you come down to it, it's just glucose. But it's put together in a very specific three-dimensional way and bonding pattern that allows for pretty good structural rigidity, structural support. It's kind of weird that simple sugar, glucose, can actually be something that's really strong. So these are representing people bonded together. You can see how they're bonded together in a long chain of glucose molecules. And they're trying to show here the all those chains of glucose get clustered together into a microfibril. All right, so that microfibril that you're seeing, all those like, tannish dots, those are all chains of glucose molecules in a big long chain. So if they're clustered together, we call them a microfibril. And then if you take a bunch of microfibrils and cluster those together, then you talk about fibrils. And so when you look at a cell wall, you can see that their three-dimensional design is not just a bunch of sugar haphazardly laid down. It's a very particular, chains of glucose clustered and then more clustering. And that is what's giving all that rigidity and structural support, even though it's glucose. All right, so cell wall structure, primary cell wall, secondary cell wall, they both have the cellulose forming these microfibrils and the fibrils. But there are other things that show up in the cell wall. By the way, if you have my PowerPoint slides up, you'll see that often there's extra notes that I've put down in the notes section. When you click on notes section in PowerPoint, you'll see that I've added some additional notes for your study purposes. So just make sure that you're aware of some of that as we work through, not every single slide, but many have that. Okay, so when you look at the cell wall, it's not just cellulose. This is trying to get at the idea that you have cellulose laid down and fibrils and microfibrils laid down, but you have other things. So pectin is being shown there down on the bottom, that neutral pectin molecule. Pectin is what you use if you've ever done canning, made jellies and jams. You probably went and bought pectin at the store to thicken up the jelly or jam. And that's what it does for the cell wall too. It's this kind of gooey matrix that allows for everything else to kind of be embedded in it. And so here you can see the cellulose. You can have proteins, or glycoprotein means it's a sugar and a protein bonded together, so sugar protein. Hemicellulose is another component of the cell wall that's related to cellulose. And so here, when you look at the cell wall, here's another image of it, where they're trying to show, here the plasma or cell membrane is on the bottom, and then they're trying to show the three-dimensional complexity of the cell wall. And then the middle lamella is kind of the cement, if you want to think of it. It's kind of like cement that sticks two neighboring cells together. So there's another, if the artist had continued drawing, there would be another cell on the other side of the middle lamella on the top here. So the middle lamella is important when you start looking at plant tissues because it keeps neighboring cells kind of stuck together. So here they're showing the pectin and the hemicellulose and cellulose. Okay, so when I was first learning what the cell wall is like, the design of it, my instructor talked about reinforced concrete, and that really made a lot of sense to me. If you've ever been to a construction site and see, you see the rebar laid down, and then the cement mixer truck comes along and dumps out all the cement, and then once it hardens up, we call it concrete. And so if you can think about that analogy, I think that's a good one with regard to what the cell wall is designed to look like when all the bits and pieces are put together. Okay. So we already saw this. So the primary cell wall has some flexibility to it, and it's thinner than the secondary cell wall. The secondary cell wall is tougher. All right, it's stronger and tougher than the primary cell wall. And it is containing an additional chemical that's unique. It's not, this chemical is not showing up in the primary cell wall. So lignin, all right? Lignin is a really complex polymer that actually they just figured out with the structure, like the actual details of the chemical nature of it, not that long ago, because it was really hard to study. And so lignin is very strong. It's part of the secondary cell wall. And so once that gets laid down, then the cell, it becomes pretty inflexible. And like I said, it also dies off. Once everything is done and good to go, then the cell will die. All right, so secondary cell walls, only some cells will get that to happen. And we'll talk more about that as we progress into the anatomy section here. By the way, if you've ever driven by like the paper company by Escanaba there, it used to be called Mead Paper. It's not called that anymore. What's it called now? Does somebody know? By Escanaba? Used to be Mead Paper Company. That's why the Mead Auditorium, there's a Mead Auditorium in the next building over. The Mead Company donated money for that auditorium. But anyways, if you've ever driven by a Mead paper producing company and smelled those weird smells, the chemical smell, they have to use really nasty chemicals primarily to get rid of the lignin. And so when you have paper that you're writing on right now, there's different qualities of paper, and the lower quality paper still has a fair bit of lignin, but the higher quality paper, like cotton bond paper or stuff up in the higher quality, they've gotten rid of the lignin. And that's the kind of paper that will get yellow with age and get brittle with age. Okay. And so lignin, it's a tough chemical, and to get rid of that as you're making paper, it's a tough thing to do. So when you think about cells that hang out near each other and they're neighbors to each other, we find that they have ways to communicate with each other in the sense of they can have flow of chemicals back and forth between neighboring cells. And so that communication that's going on is really important. And the fancy word for that, the way that they do that is through these plasmodesmata. And I like to think of them as tunnels, membrane tunnels that go from one cell to the next. And so you're seeing on the left an electron microscope picture, and those dark bands that are shown crossing between two cells. It looks like there's 4 of them here that are easily seen. 1, 2, 3, and then the 4th one down here. Those are plasmodesmata going between 2 neighboring cells. And then the artist here drew a mock-up of what that looks like. And so it's membrane lined, so it's all, kind of enclosed by this membrane tunnel system that is big enough to allow water and dissolved chemicals, small proteins, can certainly go from one cell to the next through the plasmodesmata, but bigger organelles, they're just too physically big, they can't get through. Okay? Viruses, unfortunately, can get through this way too. So once a virus gets into the plant, viruses often get into the plant through an insect vector, where the insect has it in its mouth part, and when the insect goes to feed on the plant, it gets injected, so to speak, into the plant. And then once the virus can get in, it just can go through these the plasmodesmata from one cell to the next and cause all kinds of damage. All right, so the plasmodesmata, I need you to know that term and what it's all about in terms of communication between the cells. Okay, so we'll talk more about plasmodesmata as we think about how things move around in the plant body, especially when we come to like water and things like that. So those plastids that we need to think about. So I already mentioned chloroplast, which is like super important, but there are other plastids too. And so plastids is kind of like the umbrella category. And there's different kinds of plastids. All right. So chloroplast is the most famous. Probably pretty much everyone's heard of a chloroplast that's taken some kind of biology class. Well, we need to know a little bit of the details of a chloroplast. What's it all about? So here you're seeing figure B. They're showing that the chloroplast actually has 2 membranes that surround, create that boundary for the organelle. So there's a so-called outer membrane, and then right to the inside of that, there's an inner membrane. And so sometimes they'll just say, oh, it has a double membrane. Okay, so that's what they mean by that. There's an outer and an inner membrane. And then, I don't like how they, so then all that green stuff inside, that's an internal membrane that's floating around in the watery system inside the chloroplast. So that's an internal membrane. So that internal membrane, in total, if I were to talk about the entire internal membrane, then you'd use the word thylakoid. Okay? So if I say, oh, look at that thylakoid, I'm referring to. So if it's a little artist, so let me just say this, many artists who draw for textbooks don't have biology backgrounds, okay? So that's why sometimes I have to correct some of these figures, and there's a disconnect sometimes. So if you have an artistic bent, getting, and the art and design department here is really good, getting some experience with that. I've had several advisees that do biology major with an art and design minor or double major, so they can do illustration for textbooks and things like that. It's like, yes, thank you, someone who knows biology. So the entire internal membrane is called thylakoid. Right? Now, you're going to see there's different regions of this thylakoid membrane. Some look stacked up with a bunch of coins stacked up with that one membrane stacked up. And then you can see that there's some like relatively unstacked regions that kind of look like bridges between those stacked regions. So those have different names. So the stacked regions of the membrane, those regions are called the grana. Okay, that's plural. Granum is singular, but all those stacked regions are called grana. The unstacked regions, or the ones that kind of look like a bridge, the unstacked regions are called the stroma lamellae. A-M-N is plural. Stroma lamellae. All right, so that the stroma lamellae are all the unstacked regions. Okay. So the reason they had to give them special names is that when we look at how a plant is doing photosynthesis, it's happening in the thylakoid membrane. That's why it's all green. There's chlorophyll pigment there, and chlorophyll shoots and photosynthesis. And when they look at what's happening during photosynthesis, they see some stuff happening in the grana region, some stuff happening in the stroma. So they had to give different terms so that they can talk about it as they're describing photosynthesis. Okay. And then the watery portion that thylakoid membrane is floating around in, that's called the stroma. Stroma is the watery portion inside. Then you can see on that upper figure B, you can see where the artist kind of took a cross section and cut open the grana stack here, okay? So if you cut open the thylakoid membrane and look inside of it, that region is called the lumen. Lumen, which is just referring to like in a hollow space. Like our intestines, when you look inside our intestines, we call that the lumen of our intestine. Okay, so the lumen is just the internal inside part of the membrane, if you were to cut it open and look inside. And again, certain things happen inside the thylakoid membrane, and certain things happen in the stroma on the other side of the membrane. And so again, they need to have special names. So anywhere on the you make a cut and look inside, you say, oh, I'm looking inside into the lumen. And again, certain chemical reactions occur inside like that. So they have to talk about the lumen, and then they have to talk about some of the chemical reactions that happen in the stroma, in the watery portion outside of the thylakoid membrane. Other questions on the chloroplast? Okay, so please spend time learning about everything we just talked about. I really want you to know this, because as we progress into talking about photosynthesis, I'll be using these terms. And so if you start laying that down in your neural pathways now, it'll be a whole lot easier when you get into that section. You won't be get hit with all this jargon that you haven't learned yet. Okay. Those are, by the way, figure A and figure C. Those are electron microscope pictures of actual chloroplasts. So you can see the darker gray, that's all the grana that you're looking at. And then the relatively thinner gray areas, that's the stroma lamellae, kind of the unstacked regions or the bridges. And then figure C is a close-up where they're showing a really nice granum right there, and then a couple more. And then all the whiter regions here with just a few strands, that's the stroma lamellae. Okay. So plastids. Chloroplasts are one type of a plastid, but there's others, and we're going to look at that next week in lab. For those of you taking a lab portion. So chromoplasts are another type of plastid. Chromoplasts are important in the sense that chromo is referring to color. And so we're thinking about different pigments in these plastids, and their primary goal is just to give color to different parts of the plant body. All right, so here you're looking at pepper, red pepper, and you're looking every single reddish dot that you see, that's a bunch of different plastids, or well, it's a bunch of chromoplasts with the pigment. And so when you look at that in total, look at a red pepper with the naked eye, it all looks red because there's so many chromoplasts in the cells of the pepper fruit. Here are some other examples. Whoops, sorry. Huh, I didn't label that. All right, I'll put it on the board. So on the left, well, it's down in the notes section, but it would have helped to have it on the slide. Okay, so on the left, you see a picture of a little bit of potato, white potato flesh under the microscope. All right, and it was probably stained with a little bit of iodine, I'm guessing. And so all of those kind of circular or egg-shaped looking structures, that's a type of leucoplast. Okay, so leucoplast, leuco means like colorless. So unless you do a special stain, all of these are colorless. So it does fit the category of a leucoplast, but they gave it a special name because of what it does. So you're looking at ameloplast on the left there, and it was stained with a little bit of iodine to make it pop purple so you can see it a little bit better. Ameloplast, amylo refers to starch. So these are plastids, they're technically leucoplasts, but because their function is to store starch, they said let's call them ameloplasts. That we know that, oh, starch storing. So potato tubers are a great source of starch because they're chock full of the ameloplasts when you eat them. What's the plant that we're looking at on the right? Does anyone know? I hope somebody. Yeah, avocado, guacamole. So avocados, they say that they're really healthy source of fats as long as you don't eat too much of them, right? And so under the microscope, if you take a little bit of flesh and look at the avocado under the microscope, this is what you would see. And so they store fat, and so you call them oleoplasts. Oleo is referring to lipids or fats. Okay, oleoplast. Make sure I spelled that right. Yeah. All right. So oleoplasts they're leucoplasts that happen to store lipids and fats. Okay. So like a leucoplast is like a category. It's a type of plastid, and then there's subcategories beneath that. There's the amyloplasts and the oleoplasts. All right, so different plastids, depending on the plant species and part of the plant, you might see many different types of plastid showing up. By the way, do you think it would make sense? Let's see, chloroplast. Where do most plants have their chloroplasts stuffed away and very active? What part of the plant body? Please. Would it make sense to put them in roots? No. So unless they're aerial, like an orchid. If you have orchids that, we don't have aerial orchids here. We have beautiful orchids here, but they have roots that are buried in the soil. But if you go to the tropics, most orchids live on trees without harming the tree. They just use them for support. And then they dangle their roots, and their roots actually look green because they do have chloroplasts in their roots. But the typical plant that buries its roots in soil doesn't really make sense to put any chloroplasts in typical roots because they are buried in the soil. They have no access to sunlight, can't do photosynthesis. So it just depends on what part of the plant body you're looking at and thinking about in terms of some of these internal organelles. You know, what's the composition? Mitochondria, they have a mitochondria very similar to ours. Mitochondria also have what's called outer membrane, and then they have an inner membrane, and then that inner membrane does this inhole. It's called inhole membrane, and then the watery portion is called the matrix. So right now, if you ate lunch, hopefully you did, and you have your mitochondria up and firing and making a lot of ATP for you right now, so that you can think and start learning make material. Well, plants have that too, and they want to make a lot of ATP in their mitochondria as well. They do that very similar to we do. They take in oxygen like we do. And so the reason we're taking in oxygen is basically to keep the reactions going in the mitochondria in order to make lots of ATP. So when, if you're suffocating, it's not really you're dying lack of oxygen directly. It's the fact that oxygen can't be used in this reaction to make ATP. And when your ATP levels plummet, that's when you get in trouble. Right? And it's a very quick kind of domino. Plants give off oxygen during photosynthesis, but they also need to use oxygen. So they use oxygen in their mitochondria for ATP production. It's very similar to what we do in our mitochondria. But then they also have the chloroplasts that can also make ATP. So they have this dual ability. They have mitochondria, but they also have the chloroplast. And we're going to find out that the chloroplast also generates a fair bit of ATP. Okay. And then I already talked to you about the vacuoles, and that by the time the cell has fully grown to its mature size and all of its organelles are up and running, the vacuole is the largest organelle. And again, the membrane of the organelle. So here's another nice image showing several plant cells under the microscope in that top image. And you just kind of see this empty space. Most of the internal area of the cell just looks white in this image. That's all the vacuole. It's just you don't see it unless you do a really special staining procedure. So most of the time, if you just take some cells, some plant tissue, and put it under the microscope, if it's nicely watered and well hydrated, you'll tend to see that all the other organelles get smushed up against the cell wall. And you'll see this kind of empty space in the middle. And you'll think, well, there's not much going on there. But really, it's this huge vacuole that's just full of water. And it's pushing up against the nucleus and all the other organelles. So that all of those get shoved off to the side. But that's okay because you want that turgor pressure. You want that so that the cells have that structural rigidity. So down here on the bottom, you can kind of see, we're trying to show how the vacuole it just looks like an empty space, but it's really the vacuole. And then all the other organelles are jammed off to the side. But that's creating a lot of turgor pressure in that cell, which is good. Keeps the cell up, or the plant body fighting gravity, some structural rigidity. So that's a good thing. Okay. So let's zoom out a little bit and think about neighboring cells put together in a certain way to form different tissues. So here you're looking at a mock-up of the plant body. I mean, when you think about it, they live such a different life than we do in the sense that half their body is buried in the soil. If you think about a typical plant, half their body is buried, more than half their body typically. The root system is usually bigger than the above ground in many, many plants. So half buried, half above ground. Very different living conditions. What you got going on in the soil is so, so different than having, you know, the upper body of the plant exposed to, you know, air and the atmosphere and all that. So really a different lifestyle. You can see the different key things they have labeled here. So we have the shoot apex, the root apex. So that's where you have a lot of growing going. Is length to the shoot and the root. They're trying to give you a little bit of what's happening. So that's like analogous to our blood vessel system. So they have to have some kind of internal system like that as well in order to get all the nutrients and sugars, water throughout. And then you're seeing, of course, leaves. The axillary one is a very special area that's often dormant, but if given a chance, it can start to grow and develop into branches. Then they talk, when we look at the stem tissue, there's regions of the stem that's called the node and regions that are called the node region is just simply that part of the stem where you see it. That's the node region. Then if you look at the section of the stem that's in between the in-between part, it's the internode. So the internode is that just between the two. Depending on the plant, you might have a taplet, or maybe not. You might have a taplet gland over, or you might have fibrous systems. We'll talk about that before. Okay, but that's the general. And then, of course, don't forget, we also have to think about flowers in this class of some sort. So when you think about plant tissue, it's an organized collection of cells, and they work together, they function as a unit to accomplish something for the plant. And when you get them in the right combinations, then you might start saying the word organ, a plant organ, right? So just be aware of that. So when we talk about roots, technically that's an organ, or stems or leaves, flowers. So very complex, you know, we're multicellular organisms too, and we have, you know, dedicated tissues that do very special functions for us. So it's the same idea with the plant body. They don't have lungs or liver, but they still have very important parts of tissues and organs. So when we look at plant tissues, you can kind of broadly say, yeah, there's two major categories. There's the active tissue, which is called meristematic tissue, or you might say it's that region has a meristem. Okay, meristem. So like the apical meristem. I'll show you that picture again in a minute here. But whenever you hear the word meristematic, meristem, think actively growing, like on a regular basis, day to day, hour to hour. That region of the plant body is actively growing. And when we say growing, we mean they're dividing. All right. So when we talk about growth in general in science, it means that the plant, well, in this case, the plant cells are dividing and creating new cells, or they're getting bigger. So we have to think about both. But meristematic tissue, very active area of cell division in order to add more cells to the plant body. So it definitely leads to good growth. And so that's pretty ongoing through the life of the plant. Then you have the so-called more permanent tissues that they are created by the meristematic region, but then all cells have to kind of go through a maturation process. You know, they'll grow and then they'll start forming all the organelles, and then they start taking on a special function. And so they have to mature into that functioning, mature cell. And so maybe some of those meristem cells that grow and divide are destined to become epidermal cells, which are the surface plant body. Other cells that are being produced by the meristem, eventually they're going to develop and become what we call parenchyma cells, which parenchyma cells are when you peer into the inside of a plant, body. Most of the cells that we're looking at are parenchyma cells. And so, it's all coming from meristematic regions of the plant body, but then they differentiate and form into these different tissues. Some of the tissues we call very complex. They're not simple, like epidermis is a simple tissue, parenchyma is a simple tissue. But when you start talking about like vascular tissue, which is composed of xylem and phloem, then we say, oh, those are complex tissues, just because there's several different types of cells that make up that complex. So we'll get into that in a little bit more detail. Now, those meristematic tissues, they're regions of active growth. So we find that in the apical region. So the shoot apex that I pointed out in that diagram, and the root apex, there is special regions right there in both of those areas that have meristematic growth going on. Then there's the so-called subapical region, which is just right behind the tips of the shoot and the tips of the roots, okay? Then some plants have intercalary meristem, which are between, like the best example of intercalary meristems are your lawn grasses. And that's one reason why you have to cut grass all the time. You can't just cut it once and they're done. They keep growing back. It's because even though you cut the grass short and there's just like a nub of grass left, there's an intercalary meristem in that blade of grass that will just keep growing. And before you know it, you're going to have to mow your lawn again. Okay? So intercalary meristems, we'll look at those two. And then lateral meristems are another group of meristematic areas. Lateral meristems, when you think about like, how does a tree trunk get bigger and bigger and bigger, wider in diameter? That's a lateral meristem, allowing for sometimes those tree trunks to be massive. If you've ever had the privilege to go see like the giant redwoods, giant sequoias, it's incredible. So it makes sense that there'd be regions of active growth going on to, give rise to a fully functioning plant body. And then other parts of the plant body have matured out into tissues that are like, okay, we're done growing. Now we just have to dedicate to doing what we need to do, whether it be the epidermis or the parenchyma cells. All right. Okay, we'll continue on with this on Friday and get more into the anatomy.